Hello friends, welcome back to a new video, that is MCQs on cell biology, and this is the part 4 of this series of videos on cell biology. So the first question of this video, that is, which of the following statement is correct for the endoplasmic reticulum, that is ear? So the options are option A, the SER, that means smooth endoplasmic reticulum, is the site for the synthesis, modification and shorting of proteins, or option B, that is the rough ear is the site for lipid biosynthesis, or option C, the rough ear is the site for the modifications and shorting of proteins, or option D, the rough ear is the site for protein synthesis and smooth ear is involved in modification and sorting of proteins. So the right statement about the endoplasmic reticulum is option C, that is the rough ear is the site for the modification and sorting of proteins. So this is the correct statement about endoplasmic reticulum. So let's move on to question number 62, that is, membranes of the following two organelles are contiguous, that is option A, ear and Golgi, or option B, that is nucleus and ear, or Golgi body and plasma membrane, or Golgi and lysosome. So which two organelles are contiguous or attached to each other? So the right answer is option B, that is nucleus and ear. So membranes of the following two organelles are contiguous. Contiguous is nucleus and ear. They are attached to each other by their plasma membrane. So let's move on to question number 63. That is, the endoplasmic reticulum, option A, extends from the plasma membrane into the cytoplasm, thereby creating natural entrances utilized in endocytosis, or option B, extends from the inner nuclear membrane into the nucleoplasm, thereby creating a space that is called the nucleolus, or option C, extends from the outer nuclear membrane into the cytoplasm, thereby creating a space that is separated from the cytosol. So option D, that is extends from the outer nuclear membrane into the cytoplasm, where it forms the Golgi apparatus. So the right thing about the endoplasmic reticulum is option C, that is it extends from the outer nuclear membrane into the cytoplasm, thereby creating a space or lumen that is separated from the cytosol. So let's move on to next question, that is question number 64. So which of the following statements is true for smooth endoplasmic reticulum? And the options are option A, it is made up of a single lipid layer membrane structure, or option B, it is involved in protein biosynthesis, or option C, it is involved in lipid biosynthesis, or option D, that is it is a site for interleukin 2 biosynthesis. So the right one is option C, that is it is involved in lipid biosynthesis. So which of the following statement is true for smooth endoplasmic reticulum? That is option C. So it involves in lipid biosynthesis. Okay, so next question देखेंगे. Next is question number 65. It is calcium 2 plus ATPase present on sarcoplasmic reticulum increases concentration in option A, sarcoplasmic reticulum as compared to cytosol actively, or option B, cytosol as compared to sarcoplasmic reticulum, or option C, sarcoplasmic reticulum as compared to cytosol passively, or option D, none of this. So the right answer is option A, that is sarcoplasmic reticulum as compared to cytosol actively. So calcium 2 plus ATPase present on sarcoplasmic reticulum increases concentration in sarcoplasmic reticulum as compared to cytosol actively. Okay, so next question देखेंगे. Next is question number 66. So which of the following are functions of rough ear? And the statements are statement A, or sorry, statement P is splicing of polypeptide, option Q or statement Q is formation of disulfide bond. Statement R, glycosylation of proteins during translation, or statement S, that is assembly of multi-subunit proteins. So the options are option A, P and Q statements are correct, option B, R and S statement are correct, or option C, P, Q and R statements are correct, or option D, P, Q, R and S all statements are correct. So the right answer is option D, that is P, Q, R and S all options are correct. So which of the following are functions of rough year? This all statements are correct about rough year's function. So rough year's function is splicing of polypeptide, formation of disulfide bond, glycosylation of proteins during translation, and assembly of multi-subunit proteins. Okay, so let's move on to question number 67, that is a cell which is very active in the synthesis and secretion of proteins would be expected to have option A, equal amount of smooth ear and rough ear, or option B, more rough than smooth endoplasmic reticulum, or option C, more smooth than rough ear, or option D, neither smooth ear nor rough ear. So the right answer is option B, that is more rough than smooth ear. So a cell which is very active in the synthesis and secretion of proteins would be expected to have more rough than smooth ear. Okay, so let's move on to question number 68. It is microsomal cytochrome P450 statements are monooxygenous, or statement Q, that is are membrane bound xenobiotic metabolizing enzymes, or statement R, that is they are hemoproteins, or statement S, that is they catalyzes diverse reactions such as hydroxylation, sulfoxidation, or epoxidation. So what are the right answer? And the options are option A, that is P and Q statements are correct, option B, R and S is correct, or option C, P, Q and R is correct, or option D, P, Q, R and S all are correct statements. So about the microsomal cytochrome P450. So the right one is option D, that is all P, Q, R and S statements are correct. So microsomal cytochrome P450 are monooxygenous and are membrane bound xenobiotic metabolizing enzymes and are hemoproteins and catalyzes diverse reactions such as hydroxylation, sulfoxidation and epoxidation. Okay, so next question देखेंगे. Next is question number 69. So the common precursor for synthesis of all N-linked oligosaccharide residues in glycoproteins contains a branched oligosaccharide linked to option A, KDEL sequence, or option B, GPI anchor, option C, chaperones, or option D, dolichol pyrophosphate. And the right answer is option D, that is dolichol pyrophosphate. So the common precursor for synthesis of all N-linked oligosaccharide residues in the glycoproteins contains a branched oligosaccharide linked to dolichol polysaccharide. Okay, so next is question number 70. So the oligosaccharide part of glycoproteins is either N-linked or O-linked. So the amino acid residues through which these oligosaccharides are attached to polypeptide are option A, asparagine and threonine, option B, aspartic acid and serine, option C, serine and lysine, or option D, glutamine and serine. So the right answer is option A, that is asparagine and threonine. So the oligosaccharide part of glycoproteins is either N-linked or O-linked. So the amino acid residues through which these oligosaccharides are attached to polypeptide are asparagine and threonine. Okay, so this is the right answer. So let's move on to question number 71. So which of the following is not function of rough year? And the options are option A, N-linked glycosylation of proteins, option B, folding of polypeptide chains, option C, O-linked glycosylation of proteins, option D, specific proteolytic cleavage. So the right one is option C, that is O-linked glycosylation of proteins. So which of the following is not function of rough year? That is O-linked glycosylation of proteins. This is not a function of rough year. So the next question is, which statement of the following is false statement? So option A, that is binding of acetylcholine with nicotinic acetylcholine receptor produces a net depolarization of Ru15 MV, or option B, that is neurotransmitter receptor fall into two classes, that are ligand gated ion channels and GPCR. So the option C, that is an action potential of a presynaptic axon terminal opens voltage gated calcium 2 plus channels, or option D, acetylcholine is hydrolyzed to acetate and choline by acetylcholinesterase in postsynaptic cells. So which one is the right one? Option D is the right one. So what is the question? So which of the following is false statement? So this option D is the false statement. That is acetylcholine is hydrolyzed to acetate and choline by acetylcholinesterase is postsynaptic cells. Okay, so this is the wrong one or wrong statement. So next is, which statement about the glycosylation of secreted proteins are true? And the statements are, statement P is almost all secreted proteins are glycosylated. Statement Q, that is glycosylation occurs exclusively on the NH2 group of asparagine, or statement R, that is an oligosaccharide is formed on the lipid dolicle and then transferred to the protein, or statement S, that is the oligosaccharide is trimmed by the removal of mannose residue. So which one is the correct one? So which statement about the glycosylation of secreted proteins is true? So the options are option A, that is P and Q, or option B, that is P and R, or option C, that is P, R and S, or option D, P, Q, R and S. So the right one is option C, that is P, R and S is the right one. So Q is only, that is wrong. So glycosylation of secreted proteins are almost all secreted proteins that are glycosylated. Then that is an oligosaccharide is formed on the lipid dolicle and then transferred to the protein, and the oligosaccharide is trimmed by the removal of mannose residue. So these are all correct options. Okay, so next question is, which one of the following statement is not true about protein glycosylation? And the options are option A, protein glycosylation is one of the major biosynthetic function of smooth air, or option B, that is it is a process of covalent addition of sugars to secretory proteins, or option C, that is an oligosaccharide containing 14 sugars is transferred to the NH2 group of asparagines residue of a protein, or option D, that is antibiotic tunicamicin inhibit the first step of end-linked glycosylation. So the right one is option A, that is protein glycosylation is one of the major biosynthetic functions of smooth air. So which of the statement is not true about protein glycosylation? That is option A. So option A is not correct. Okay, so next question देखेंगे. Next is, during N-glycosylation, oligosaccharide is added to asparagine residue in the consensus sequence of option A, that is asparagine, then X, then serine or threonine, where X is only amino acid, X is any amino acid except proline, or option B, that is tyrosine, then leucine, or option C, that is tyrosine XX serine, where X is bulky hydrophobic amino acid, or option D, leucine phenylalanine. So the right answer is option A, that is asparagine X serine or threonine, where X is any amino acid except proline. So during N-glycosylation oligosaccharide is added to asparagine residue in the consensus sequence of asparagine, then X, then serine or threonine, where X is any amino acid except proline. Okay, so next move on to question number 76, that is choose the statement that is not correct for Golgi apparatus of the cell. And the options are option A, degradation of xenobiotic compounds occur in Golgi apparatus, or option B, Golgi apparatus has cis and trans phases, option C, that is Golgi apparatus is predominant in cells that are specialized for secretion, or option D, that is it is involved in oligosaccharide modification. So the right answer is option A, that is degradation of xenobiotic compounds occur in Golgi apparatus. So choose the statement that is not correct for Golgi apparatus of the cells. So option A is not correct about Golgi apparatus. Okay. So next move on to question number 77. That is, which statement is false? So the options are option A, the Golgi complex forms vehicles that fuse to form the endoplasmic reticulum. Option B, if a lysosome burst, its content can seriously damage the cytoplasm of a cell. Or option C, secreted proteins are initially formed by ribosomes attached to the endoplasmic reticulum. Or option D, the nucleolus is where ribosomes are assembled. So the right answer is option A, that is false statement. Which one is the false statement? That is option A is the false statement. So the Golgi complex form vehicles that fuse to form the endoplasmic reticulum. This one is the false statement. So let's move on to question number 78. That is nicotinic acetylcholine receptors, and the statements are statement P is nicotinic acetylcholine receptors expressed in muscle cells is a ligand gated channel, and statement Q, that is present nicotinic acetylcholine receptor present on postsynaptic membrane, or they contain alpha subunits that bind to the acetylcholine, or statement S, that means this receptor allow passage of both sodium and potassium ions in hydrated form. So which combination of statement is correct? So options are option A, P, Q and R is correct, option B, P, Q, R and S is correct, option C, P and R only, or option D, Q only is correct. So which one is the correct statement? So option B is the correct one. So P, Q, R and S, all these statements are correct about nicotinic acetylcholine receptors. Okay. So next question देखेंगे. Next is question number 79. That is fibroblasts do not generate an action potential because of absence of voltage gated ion channel. Option B, there is no sodium potassium pump. Option C, their membranes have large number of porins. Or option D, their voltage gated sodium ion channel work at very high rate. So the right answer is option A, that is of absence of voltage gated ion channel. So fibroblasts do not generate an action potential because of absence of voltage gated ion channel. Okay. So the last question of this video, it is which one of the following is not a function of Golgi apparatus? So the options are option A, that is proteins synthesized by the ribosomes on the rough ear are transported to Golgi where it is accumulated in the sacs, or option B, that is protein filled sac may be retained within the cell as lysosome, or option C, it is the site where glycosylation of lipid takes place, or option D, it is the site where synthesis of phospholipid takes place. So which one is not a function of Golgi apparatus? So the right one is option D. So option D is not a function of Golgi apparatus. So it is the site where synthesis of phospholipid takes place. So this is wrong one. Okay, so these are all 20 questions of this part 4 video on cell biology MCQs. Okay, so thank you for watching this video.